Grateful to the Society for Reproduction and Fertility for their generous sponsorship of this series. So I'm going to talk to you today about my favourite topic, which is pregnancy and the processes in reproduction that involve the immune system. And you may say, why is the immune system important in reproduction? Well, when you think about it, the fetus gets its genes 50% from the mum, but 50% from the father. And that means that when the embryo implants, it's effectively foreign to the female body, and theoretically should be being rejected in the same way as a heart transplant or a kidney transplant is rejected. Why this does not occur is one of the most fascinating questions in biology, and has really consumed immunologists and reproductive biologists for decades to try and sort it out, and in fact is critical, we now understand, to understanding where the origins of many of the disorders of infertility and pregnancy that lead to preterm birth, fetal growth restriction, and some other conditions of pregnancy that really set the life course health in all of our children. So the immune response to reproduction and pregnancy is one of the most important things we can study and one of the most fascinating, and I hope you'll get a sense of that from the stories I'll tell you today. But before I start with that, I want to just say a little bit more around why reproduction science is so important. Reproductive biologists often hear, oh, this isn't the most important area of biomedical research. It isn't a disease that we die from. In fact, isn't it the case that we have too many people on the earth? And that's the major cause of all of our problems. Well, as it turns out, things are changing. And while we have had an enormous phase of population growth, for the first time ever in the history of our Earth, our population is looking like it's going to turn around and go into negative territory within the next couple of decades. So this is a map of the fertility rate in each of the countries of the world, and we know we must have at least 2.1 children per woman just to sustain the population that we're currently at. Well, 50% of the countries in the world, all of those that are coloured red or pink or pale blue or purple, are below replacement rate. And if it was not for the case of Central Africa, where the reproduction rates are much higher than the rest of the world, we would already be in negative territory in terms of the world's population. So we now have a situation for the first time where many countries, especially Japan, Germany, Singapore, even China, is becoming really concerned about the future of the demographics of those countries, and is expecting within, for example, in Germany, the next 40 years to have a 30% lower population rate. And the only reason we haven't hit that territory yet is because people are aging, and so we've got this very dramatic change in population demographics. But as older people begin to die, we will be left with a situation where we will be below the 1.7 critical rate at which continuing decline of the size of the population is projected to be unstoppable if it continues in that way. So I reckon that reproduction is going to be in the headlines, and the fertility rate increasingly in all of our countries over the next several years, including in the UK, where already this is in the public discourse. And people are very concerned that we're now in this country below or at 1.7. Last year it was at 1.8, so well below that replacement rate, and looking to a trajectory of declining population in the near future. Nearly half of all countries are now facing this baby bust, meaning there are insufficient children to maintain population sizes. And this shocks people. They're used to hearing that we have too many people, but when you consider how we're going to cope in the future in 20, 30 years' time with very few children and very large populations of older people, we're going to be wondering how indeed to improve reproduction and fertility. And China is already discussing what this will mean for their demographics, where the expectation is within 30 years they will have 200 million workers missing. And the impact of that on the demography, the economics and business and political impacts are going to be enormous. So even as little as 10 years ago, we were projecting that this beginning of levelling off in the world's population would occur towards the end of this century. It now looks like it's going to be by around 2050. Questions around why our fertility rate is so low will be occupying all of us. And clearly there are many reasons, and several of those are social. They're to do with education of women, availability of contraception, and that's really important for bringing down fertility rates, especially in countries like Africa or the countries in Africa where this is the cause of some other difficulties. So we need to bring down population rates, but not to the point below which they're irreplaceable. So social factors are a major cause, socio-economic factors, including in Western countries where people are making different decisions and then getting to the point where they aren't thinking about having children until their later 30s is critical. But as well as the social factors, there are biological factors. And this is the best example of that. A major paper that was published around just over a year ago, where a series of what we call meta-analyses were brought together with some 40,000 men across most of the Western countries in the world, showing that male sperm counts have declined by half in the last 40 years. Now, we can't invoke a social reason to explain that. That's about our environment, something to do with the modern 21st century, indeed 20th century environment, impacts of a range of exposures. We can't explain this with genetics. It's about environmental impact on our biology. It's affecting our animals. It's affecting our insects. And it's likely to be a combination of a series of things: sedentary lifestyle, poor diet and nutrition, metabolic dysfunction and hyperglycemia, exposures to environmental toxins, chemicals and plastics, herbicides and insecticides are a major contending factor, possibly unresolved sexually transmitted infection. So these are the things that biologists, reproductive biologists, are now grappling with and building the evidence to convince our governments and communities that we need to really make a difference if we want to otherwise divert the consequences of this baby bust. Other people tell us, well, we've got the solutions, why are we worried about it? Surely we can fix it with IVF? Well, that's not the case. IVF might be thought of by many of the world's population, the 1 in 6 couples, or over 15%, that's 50 million couples around the world, to be the way that they might solve their infertility problems. But in fact, it's not a cure, it's just a workaround. And importantly, only around 50 to 60% of couples who seek help with IVF are likely to have a healthy baby. It's expensive, it costs individuals and governments and societies large proportions of their public health purse, and it shouldn't be costing that. It's invasive, stressful, disruptive. It causes major anxiety and has impacts on women, men and families. And importantly, it has health risks for particularly offspring, but also women who are exposed to different IVF interventions, and often these are also not well discussed. Infertility is just the tip of the iceberg. So for the 1 in 6 couples who have trouble struggling to conceive, of these 100 couples I've got sketched out here, another large proportion of them will conceive but will result in miscarriage, and in a smaller proportion, recurrent miscarriage, so 3, 4, 5, 12 or 14 sometimes miscarriages, and you can imagine the anxiety, the anguish indeed of experiencing that. Then there are other couples who progress to what looks like a healthy pregnancy, but later in gestation will struggle with conditions like preeclampsia, where the placenta isn't working as well as it should to deliver the nutrients to the baby. There might be fetal growth restriction, where the baby isn't growing as well as it should and has to be delivered early, or what we call spontaneous preterm birth, where the baby comes of its own accord, but too small and too early. And then there is a proportion of pregnancies where the fetus is developing with a genetic or a congenital abnormality. So the reasons why we're so concerned about those pregnancy disorders is that they obviously cost our women's and children's hospitals enormous amounts of money that have an impact on that infant for the several months, often after it's born, and the families. But it isn't just in that period around the time of birth. Children born with less than optimal experiences in their pregnancies go on to have lifetime consequences. And this is because of a whole area that we're now coming to understand called developmental programming, where we understand that the experiences of the fetus during vulnerable stages of development before birth set the course of development to influence the phenotype of the offspring. So establishing the metabolic function, the immunological function, neurological structure of the tissues and the function of those tissues, how they'll operate in that individual after birth. And that can have a lifetime legacy. And it's particularly the case for these babies that are born too small or too early, where many of the non-communicable diseases that we're very concerned about because they're on the rise are particularly impactful. So the metabolic disorders that lead to later life heart disease and diabetes, neurological behavioural disorders, autism, memory and learning difficulties, allergy and asthma and other immune disorders, all of these are increased, particularly in babies that have had difficulties before they're born. But now we also know, even in children who would otherwise be, by all means, looking like healthy kids, those that have higher rates of these conditions likely had events before they were born that began to set the seeds of setting their susceptibility to maybe a second hit or another insult, which then increases the chances of them having those conditions. So I'm trying to set the scene here by saying that I can't emphasise the importance of understanding the health of pregnancy and the reproductive contributions to setting our life course trajectory and the health of all of us in our communities, and the importance of focusing on this very early part of life if we're serious about doing a better job at prevention of these conditions and having healthier communities to live in. And that brings me to thinking about the immune response, because when we ask what are the factors that impact how a baby develops before it's born, and indeed whether or not a couple are fertile in the first place, whether an adequate implantation will occur and the placenta will develop in such a way as to sufficiently nurture and deliver enough nutrients to that fetus, all of this is impacted by several things: our genetics, nutrition, environmental exposures, but particularly it's impacted by the immune response in the mother. So the way the female body is dealing with those foreign tissue antigens, the molecules that are expressed by the father's genes that are present in the fetus itself, but also importantly in the placenta, the part of the tissue that's interacting with the mother's body, that immune response is really identified now as one of the key drivers of the health of establishing the pregnancy and ensuring that the baby will have the best chances after birth. I'm sorry my slides are slightly cut off over there on that left-hand side, but what the understanding of the immune response and its paradox, I guess that we call it in pregnancy, is all about this issue of how it is that a fetus, despite expressing genes from the father that should make it foreign to the mother's body, how it is possible that the female immune response is not rejecting that fetus, but instead is nurturing it and allowing it to invade and be supported and develop to term is a very important question. And I'm going to deal with that question in four areas of discussion. First of all, explaining what it is that we know about the immune response to conception and pregnancy. A little bit about the biology of particularly the interaction between not just the mum and the baby, but the father's contribution, and how this is really about the triad, if you like, of the genes of the mum, the dad, and the baby. And that's where this issue of compatibility, that interaction between those genetics, is important. And then I'll comment in the last section about how this is relevant to human reproductive health. So, as I've tried to explain, the understanding how these two systems are working together is an enormous question. Our immune system should all be about, we often think about recognising things that are foreign, defending our body from them, rejecting things that are invaders, and our reproductive system is about accepting tissue that is foreign, nurturing and supporting it. So how to reconcile how it's possible these two systems could be interacting. So the first scientist who really dealt with this question and articulated the issue was Peter Medawar, who's well known as the grandfather of transplantation immunology. And indeed, in the 1950s, Peter Medawar said, how is it that the immune response could possibly be coping with that foreign tissue during pregnancy? And he thought that the explanation must lie in thinking either about physical separation of the mother's immune system, so it never came into contact with the foreign tissues of the embryo and the fetus, turning off the expression of those genes. So the genes from the dad might be there, but the proteins, the foreign proteins, might not be actually made. Or thirdly, he thought there might be some kind of systemic turning off or silencing of the immune response in pregnancy. Well, as it turns out, none of those three alternative hypotheses are a sufficient explanation for how immunology of pregnancy is possible. And it turns out, in a way, they were kind of the opposite, because, in fact, what we now understand is that the mother's immune response is really active in pregnancy. It's really aware and responding to those foreign male transplantation antigens expressed by the fetal tissues, and indeed is intimately involved with and contributing to the events of reproduction in pregnancy in a very positive way. So how is that possible? Well, it's come to be explained by an understanding that the immune system isn't always about rejection. So in the last 20 years or so, immunologists have really figured out that indeed there's two parts of our immune response. There's the immune defence and rejection that we come to be familiar with in undergraduate training, but also there's the other side, which is all about tolerance. So accepting things that, despite being foreign, are not dangerous to our bodies. So the immune system has to recognise and then discriminate between challenges that are dangerous, and I've drawn here anthrax infection or a tumour, and alternatively things that are foreign but benign, that are not difficult for our, or a challenge to our function. And for example, the bacteria, the friendly bacteria that live in all the mucosal tissues of our body, and the many foreign materials that we ingest all day long, and the food we eat, the air we breathe, the materials we put on our skin, all of these stimulate an immune response in our bodies, but generally they are not immune rejection unless there's problems with allergy. Generally they are tolerance. And it's this kind of immune response, this immune tolerance, that's important in the reproductive setting. Now, the way that can work is that there are a bunch of different kinds or flavours of immune cells in our immune system, and we have been familiar for many decades for the ones that are to do with killing and rejection and defence, and they're drawn here in the pink and red colours because they're dangerous to pregnancy. But then on the flip side, we have these other immune cells that are all involved with recognition, but then supporting and enabling and regulating homeostasis and managing immunological tolerance. And the cells that are really critical for that are especially these regulatory cells called Treg cells, macrophages and dendritic cells, which stimulate Treg cells to be tolerant, and some kinds of, indeed, natural killer cells, even though they have the name that makes them sound rather dangerous. Uterine natural killer cells, on the whole, are supportive and enabling of pregnancy. And we can see that there are enormous numbers of immune cells in the reproductive tissues. This is mouse uterus stained with a bunch of different antibodies to recognise different markers on immune cells. And it's just simply to show that they're a very heterogeneous population. There are many different types of them there, but enormous numbers of them there. They make up to about 20% of the cells in the reproductive tissues and in the surface of the uterus. So they're very prevalent and clearly give immune competence to this tissue. And we can show that there is an active immune response in pregnancy in different ways. This is a mouse uterus. The mouse has been opened at autopsy in late gestation, and you can see these different little, like peas in a pod. These are the fetuses in the litter of the mouse in late gestation. Each of these, inside of it, will have a fetus and a placenta. Placenta doesn't come fluorescent green, it's just coloured that way so you can see it. And importantly also, what we can see here is that there are lymph nodes. These are the para-aortic lymph nodes that drain the uterus. And when the mouse is pregnant, they increase in size by about 2 or 3-fold, reflecting the proliferation and activation of immune cells in that tissue. So clearly showing us there's active engagement and proliferation and involvement of immune cells responding to those foreign tissue transplantation antigens on the fetus. Now, if we go a bit deeper into the tissues, and this indeed is a photograph from the uterus of a pregnant woman, and we can see now that this tissue, which is taken from the site where the placenta is interacting very closely with the mother's uterus, the part of the uterus called the decidua, the salus, and look inside there, there are trophoblast cells, which are the most invasive cells of the placenta, and these darker brown cells are the mother's immune cells. And see how closely they are interacting with and engaging with those foreign trophoblast cells derived from the placenta, expressing the father's transplantation genes. So clearly there's very close interaction and engagement going on here. So the way that this is all possible is that it works because the immune cells that are present are all of those immune cells that are the friendly, tolerogenic immune cells in a healthy pregnancy, dominated by Treg cells, natural killer cells, macrophages and dendritic cells, all supporting and nurturing the tissues. And the way they operate is to prevent the generation of the bad guy, orange and red rejection cells, and to suppress inflammation in that tissue. And indeed we can show this in another way. There is a huge inflammatory load associated with pregnancy because of all that foreign antigenic information. There's also a lot of tissue remodeling and tissue reorganisation going on that normally would cause almost a kind of injury or a stimulation to the tissues. And that inflammatory load is only possible to be sustained over the course of gestation because of an equal and opposite force that's coming from these anti-inflammatory cells, and especially those Treg cells. And what I also want to show here is that being able to keep that inflammation under control has to occur right from the very beginning, when the embryo is first beginning to implant at the time, just in the days or first week after conception. So it has to get generated really early on. But as the inflammatory load builds and the fetus is getting larger and the tissue is getting more and more stretched and there's more and more challenge because of the weight of all of that antigenic load, the anti-inflammatory protection starts to wear out, and ultimately this leads to a shift in the balance and the immune response tips over into inflammation and starts the process of parturition and preparation for birth. So we need those Treg cells in particular, and we need them from very early on, and we can show using animal studies that if we delete them, and we can do that with genetics or using special antibodies, that if we need them right from the moment the embryo is what we call the blastocyst stage. So here it's about 100 cells big in the mouse. This is only about 3 days after conception, and in the next hours that blastocyst will break out from that zona pellucida and begin to attach and outgrow and invade into the surface of the uterine wall. Now, if these immune cells are removed before that embryo begins to implant, there'll be complete implantation failure. And the cells that are most critical are Tregs. If we take out macrophages or dendritic cells or natural killer cells, the pregnancies aren't so great, but they do continue. Whereas if we take out the Tregs, it's finished. They are absolutely essential. If we take them out a little later, once the implantation is commenced, then we can show that they are also important during placental development. And if they are insufficient or taken out experimentally, it will increase the likelihood of miscarriage, and they're also important to protect against later gestation loss and preterm birth. But the time they're most critical is at this very early time. And the other thing that they're doing, and the reason that they are so linked with that implantation success, is that they are assisting the placenta to develop. And they do that by especially talking to all the other immune cells in that decidual surface to help the blood vessels relax and get a sufficiently large diameter to allow the blood supply to come through to bathe the surface of the placenta trophoblast surface. And that blood supply is so critical because if it isn't sufficient, the fetus isn't going to get enough nutrients. And that becomes a problem particularly in later gestation when the fetus is getting much bigger. And if these blood vessels haven't relaxed enough in the beginning of pregnancy, that's what makes susceptibility to preeclampsia and in utero growth restriction much later on. So the events that are getting this established at that early time are not just allowing implantation and protecting from miscarriage, they're setting the course for the likelihood of problems that may otherwise arise in later gestation. So in summary then, Medawar's idea that the immune system is somehow turned off or physically separated is wrong. No, this is not what's going on, and instead there has to be an active, competent immune response. But that immune response has to be biased towards immunological tolerance to allow healthy pregnancy to occur. If there isn't sufficient direction of the immune response towards tolerance, and if instead you're getting a shift towards that a more immune defense kind of response, and those cells that are in the red and orange colours, then that is contributing to these disorders of pregnancy that I've described. And we now know that for infertility, recurrent miscarriage, preeclampsia, and even preterm birth, each of these is linked with different degrees of impairment of generation of this immune tolerance. And in each case, the Treg cells have been implicated and shown to be different in populations of women that experience those different conditions. And the extent to which they are the cause of all of those conditions, I think, is still debatable, but they clearly are involved in some way. So what's going on to set the course of that healthy immune response? Well, it turns out the events at conception are the most critical, because this is when things have to get established in the first place. So right from the time the embryo is first being formed, at the time of conception, when the sperm are fertilising the egg and the first divisions are happening, that blastocyst is developing and getting ready to implant. At the same time, the uterus and the immune adaptation to generate these Treg cells is occurring. And the conception phase is really initiating the trajectory of that immune balance and determining the strength of that anti-inflammatory tolerance. And if things aren't going right at that conception phase, that then will mean there won't be enough tolerance to last the course of gestation, and this inflammatory load will build too strong. So what's going on around conception to set the scene in the right way? And that's where the animal models are really helpful to us, because you can imagine it's really difficult to sort this out in human clinical tissues. So we've used mouse models a lot, but there's been similar work done in other large animal species. And what it shows is when the embryo is getting ready to implant, already these immune cells are present in the endometrial or the surface of the uterus. especially these Treg cells, have to be there. And the really interesting part that we've discovered in our laboratory is that critical for getting them there is the father's contribution, and especially the seminal fluid contact that happens around the time of conception and in preconception cycles is stimulating the immune response to get prepared and get primed in the right way to have these Treg cells present. And this is telling us that sperm is more important than the single sperm that fertilizes the egg. It's got another job in the female reproductive tract, and that's about immune education or priming of the female immune response. Now, it actually isn't surprising when you consider that there's something like 10 to the 9, I think, sperm in the average human ejaculate. You think, well, how come if we only need one, we need all those others? Well, all those others are talking to female reproductive tract cells, many of them. And you can see here how closely they attach to and potentially are communicating with the cells lining the surface of the uterus and the oviduct, and this is actually human tissue with human sperm. These higher magnification electron microscope slides show us that the sperm are lining up. These are the sperm heads on the surface of the epithelium, lining up there against these little fimbriae of the epithelial cells, and some of them are even embedded inside the epithelial cells. And in fact, if we use mouse studies and label sperm in the male with special high molecular weight hydrogen so we can track it, we can show that about 10% of the nucleic acid from the male sperm after mating in the mouse finishes up in parts of the female body that are external to the lumen of the reproductive tract. So they're crossing the surface of the epithelium, they're taken up in immune cells, and they finish up in lymph nodes, in the ovary, even in the adrenal gland. So sperm are doing a bunch of things other than just fertilizing an egg. And it's not just the sperm, but the soluble material between the sperm, the plasma fraction, contains a bunch of signaling proteins and other molecules that have biological activity in talking to female cells. And there are special signaling molecules that we've identified. One of them is transforming growth factor beta, TGF-beta. There are prostaglandins, which are smaller molecules, and also these nucleic acid messenger molecules called non-coding or microRNAs. And they can alter together the gene expression profile of the female tissues after the coital activity. So both the sperm and the seminal plasma fraction go into the female tract and activate an altered gene expression profile with some 2,000 genes differentially either upregulated, mainly some downregulated in these female cells. And that's the first step in turning on the immune response. And we can see that immune response happening within 12 hours. If we look at the mouse uterus now, here we are, the 12 hours after mating, and all these golden brown cells are immune cells, and they're mainly cells that are involved in stimulating a new immune response, what we call macrophages and dendritic cells. And they last in this kind of inflammation-like response for about another 72 hours before moving to the lymph nodes and activating T cell changes. And the consequence of that, if we look at the T cells in the lymph nodes, is that the numbers, especially of the Treg cells, increase about 2 to 3-fold. And that's happening in the lymph nodes, where I showed you that drain the uterus, and then those cells leave the lymph node, traffic through the blood, and are collected back into the uterus to be there ready when the embryo is getting ready to implant. But it turns out you need both the sperm and the seminal plasma fraction of the ejaculate. If we either vasectomize the male mass to remove the sperm, or we excise surgically the seminal vesicle glands where the TGF-beta is made, that removes that capacity to elicit this immune tolerance response. There's also all these microRNAs that are being delivered and or turned on in the female tissues, and they're also assisting with changing that female genetic expression. And we can see them here in this way of specially looking for microRNAs, that they are turned on after the sperm contact the female reproductive tract in those purple spots. Now, the consequence of all of that is that it induces these Treg cells. But we wanted to know, well, what happens if the mice have sex more than once before they get pregnant? Because that's pretty much what humans are doing, right? So after one coital activity or mating event, the Treg cells have increased 2 or 3-fold, like I said. But here they are. If there's 2 mating events with the same male partner, they're further increasing, 4 mating events increasing again. So this is showing us, like any other immune response, really, you have a priming and boosting effect of regular exposures, but it has to be to the same male partner or the same antigenic information to get that response. So how important is this? If we take away these signals from the male and commence a pregnancy without them, is there a consequence? And we can look at that by using a model where we surgically remove the seminal vesicle gland so that there is no seminal plasma, but there's still sperm, so the mice will get pregnant. And what we find in that case is that the consequence of the pregnancies is that they're severely affected. There's many fewer of the females that actually get pregnant, but those that do have a smaller litter size. A larger number of the pups in the litter don't develop properly and die during gestation. And those that are born, apparently looking okay, then develop problems into later life. And they, particularly as adult offspring, become obese and experience high blood pressure and other signs of metabolic disorder as a consequence of just having been conceived in this environment where they weren't getting the right signals from the father at the time of conception. So this tells us that sperm is more important than just fertilising an egg, and it can really have an impact on the way a fetus develops and the outcome of a pregnancy, not just by affecting the embryo, but by affecting the female reproductive tract tissues in order to get that preparation for the pregnancy happening correctly. So to what extent is this compatibility idea contributing? Compatibility is the idea that there may be better combinations of different males and different females to elicit a stronger kind of immune response. So is it important? Well, if we look in pretty much any species, either a mammalian species or invertebrate species, where there's intromission, delivery of male seminal fluid to the female tract before the pregnancy or the conception, there is evidence of an immune response occurring. So that happens right from Drosophila through all of the mammalian species that have been examined to varying extents and in different ways and forms, and there can be a bit of difference depending on the reproductive strategy of the different species. But pretty much all of them have an immune response to conception, including, as it turns out, people. So why was it important? Why was Medawar wrong, in fact? I mean, we might have thought that if the immune system can be difficult in pregnancy, and as it is in some of these pregnancy disorders, it might have been safer if we just evolved, like Medawar suggested, to keep the immune response out of it. Well, the fact that the immune system is involved and is so intimately involved means there must be some benefit, some biological and evolutionary value of immune regulation of conception and fertility. So why is it that a choosy immune response is more advantageous than none at all? Well, I reckon it's to do with maximising the chances of healthy offspring and indeed maximising the chances of outbreeding. So the concept that we need outbreeding and to prevent inbreeding is familiar to most animal scientists and veterinary scientists who know about hybrid vigour. But it's also the case in human populations that we need to ensure that we have sufficient genetic diversity, and particularly diversity in our immune response genes. Now, genes that seem to contribute to maximising this immune diversity are what we call the MHC, sorry that's been cut off, the major histocompatibility complex, or MHC genes. And the idea is that if you mate two of any species, they could be mice, they could be humans, I've drawn butterflies, if they've got the same MHC, they're going to have fewer offspring than if the parents were MHC disparate. So it maximises the chances of fertility and of a larger reproductive outcome and of healthier, more robust offspring. So it turns out that many species have a series of different systems for ensuring that they maximise outbreeding and genetic diversity. And it starts with pheromones. So animals have different ways of sharing information about their MHC based on sharing pheromones, and they can do that by exchanging biological fluids in different ways. Humans do it by snogging. So we make decisions based on who we prefer as a mate depending on MHC, initially using pheromones. And so pheromones are part of the pheromone sensing system is partly encoded by these MHC molecules. But I reckon there are also, and there are others now who've shown, there are also mechanisms that occur after mating when the seminal fluid is delivered in the female tract that are sensing the MHC and making decisions about female reproductive investment. And we can show with Drosophila that the chances of sperm competing out other sperm and successfully conceiving are higher if that male is MHC different or disparate to the female. So this is a process known as cryptic female choice. It's the idea that the female reproductive tract has this hidden capacity to make decisions and to respond and invest in reproductive activity depending on how the male gametes are evaluated. So it's discrimination of the quality of the opportunity, the reproductive opportunity for reproductive investment, and the capacity to either support or turn off progression accordingly. We can see it in mice. This isn't the easiest piece of information to digest, but basically, if the mating pair is genetically identical, there are poorer outcomes than if the mating pair is genetically identical in every gene other than the MHC, right? So we can compare the importance of just the MHC being different and show that you get higher fetal weights and a higher fetal placental weight ratio with a better efficiency of the placenta if the MHC is different. And it seems to be correlated to this business of affecting the blood supply into the placenta. So when there's different MHC, there's larger vessel diameters in those decidual blood vessels delivering nutrients into the placenta, which supports a stronger pregnancy and a healthier fetus. So what this means is that the immune system is working with the breeding process or the reproductive process around the time of conception to ensure that there's greater diversity between males and females, or that is preferred. So for a female, the greatest diversity is achieved if she comes across a male that isn't the same MHC, but instead one that's different. And indeed, the greater that MHC disparity, the better. But the other critical part of this is that the strength of her immune response, so her ability to distinguish and respond to that MHC difference, is a preferential factor. So if there is a poor immune response or insufficient stimulation of her immune response from the disparate male, that won't be as good as this case. So that is the preferred outcome. The greatest MHC disparity and the strongest MHC signal gives the greatest compatibility from a reproductive perspective. So how and why did this evolve? Well, it's about maximising reproductive success. Females have limited reproductive opportunities, unlike males, where the strategy is usually to send the sperm as far and wide as possible. The female has to make sure that when she invests her gametes, she's doing it with the right partner. She must ensure the best investment of her reproductive resources and favour pregnancy with a compatible partner and avoid pregnancy when things aren't quite right, when the male gametes aren't the best ones, they might be low quality or incompatible immunologically. And the same processes will assist in ensuring fetal integrity and developmental competence. And alternatively, and as well as when environmental conditions are not appropriate. So if there are insufficient resources, nutritional resources, or a stressful environment, the immune system can also be co-opted to operate in a similar way. So our hypothesis that we're currently working with is that this whole immune engagement at conception is to do with an immune-mediated quality control, if you like, that is equipping the uterus with a sophisticated quality control system that integrates compatibility signals from the male seminal fluid, the embryo, the placenta, but also with external and internal environmental signals, and discriminates good from bad reproductive opportunities, then acts to support or suppress progression of that process. So you can imagine how this would be operating at a population level to maximise outbreeding and to ensure that we maintain healthy populations. It's more of a problem, obviously, at an individual level. If you've got a situation where you want to have a pregnancy with a particular person and it's not working and you think there's a compatibility issue. So, of course, understanding what's going on here and getting to the nuts and bolts of all of it will help us, I think, figure out how to work with the process in the best possible way, but also deal with it when it's not working in the way that it should be. And together we can see then that the immune and reproductive systems are interacting and are helping each other, and at a population level this has evolutionary benefit because the immune system wants to be as diverse as possible, so when we get the next infectious disease, the bird flu, might knock over 98% of us, but if some of us have got the MHC genes that will allow us to reject that infection, the likelihood of the species surviving is higher. So at a species or population level, this is important. The reproductive system is helping with that and is using the immune system to maximise the best reproductive outcomes and the greatest health and diversity in our offspring. But is this working in people? Well, I've only got 5 minutes left to tell you. There is evidence that indeed similar processes operate in people. And of course, it's much harder to do the science to understand it, but we do have evidence that the elements of this response are present and are operational in our species. So some of the evidence comes from studying large numbers of populations, and especially comparing populations where there's lots of outbreeding compared to populations where there's more likelihood of people intermarrying or having what we call consanguinity. And what we find then is if there is sharing in these MHC genes between the mother and the father, the chance of becoming pregnant is lower, it takes longer to get pregnant, and miscarriage is more likely when there is shared MHC genes between the mother and the father, especially in the MHC genes that are called HLA-C that are expressed on the placental cells. And this may explain why we have such a large degree of loss of conceptions in people. Humans often get accused of having pretty poor reproductive capacity, and we're told that the fact that we lose about 50 or 60% of the embryos that they never implant and begin to form a pregnancy is because we're because we're reproductively not very good. But I would argue that in fact this can also be interpreted as evidence that there's a really active selection process going on, a kind of quality control system or process that's evaluating the opportunity, the gametes, the embryo, and making sure that things only progress when things are set up in the best possible way. We also know that all the elements of the communication between the male system and the female system are present in humans. So in male seminal fluid, the signalling agents are all present. These MHC molecules are present in a soluble form, the TGF-beta, the microRNAs and the prostaglandins are present. Interestingly, also, we find that as well as these permissive molecules that are persuading the system to become tolerant, there is also sometimes presence of molecules that interfere or inhibit generation of immune tolerance. So particularly when there's sexually transmitted infection or when there's other inflammatory stressors in men, we've been able to show that the increase in gamma interferon in the seminal fluid is linked with a higher chance of infertility. And we're working with these clues about measuring the balance of these different permissive and inhibitory molecules to develop new tests for male fertility that would go alongside working with sperm counts. We also know that when women have intercourse and their partner is not wearing a condom, they have the same kind of immune response in the cervix. So we were very pleased to be able to work with our collaborators in Sweden to take these tiny biopsies from the cervix before intercourse and after intercourse, and to find that the immune cell response in the cervix in women is very similar to what we described in other animal species, with all these immune cells coming in. And you actually need the contact with the seminal fluid to make this happen. If the couples are wearing a condom, the actual act of having intercourse isn't enough. You need that seminal fluid stimulation. There's other evidence that comes from clinical studies that shows that contact with the conceiving partner's seminal fluid is beneficial. Having intercourse around the time of IVF embryo transfer improves the chances of getting pregnant. Couples who get pregnant after a very short phase of having sex together are more likely to have problems that have an immunological basis. In particular, they're more likely to have this condition preeclampsia, where their T cells aren't working correctly and the baby is born often too small and too soon. Immediately switching from barrier contraception to becoming pregnant can also be linked with an increased rate of preeclampsia, and you can imagine that that's because there hasn't been contact with the seminal fluid before the conceiving event, compared to people who are using contraceptive pill or other approaches to managing their fertility. In IVF treatments, couples who use donor gametes, in particular donor oocytes, seem to have difficulty with this priming and have a higher rate, particularly of preeclampsia. So it's about 30% chance of getting preeclampsia with donor gametes compared to 3% in the rest of the population. And that's consistent also with the fact that in that case, there hasn't been this priming with those foreign antigens before the conception begins. The evidence that seminal fluid contact improves IVF outcomes is very strong. It comes from a large meta-analysis with about 8 studies included, about 2,000 people altogether, showing a 24% increase in pregnancy rate simply by having intercourse around the time of embryo transfer. And many clinics are now including that in their practice. So it's telling us that in humans, as in animals, that the seminal fluid contact is exerting an impact on the reproductive process beyond simply delivering the sperm to the egg, and that seems to be linked with this immune adaptation and benefiting outcomes. We think that there is learnings to be had here about understanding what goes wrong in infertility. We can show that the variation in the levels of these factors, either the TGF-beta or the gamma interferon, is quite highly variable, even within men, but also between men, where we can link the relative balance of these factors with the likelihood of fertility, and especially this high gamma interferon seems to be problematic, and we reckon that that might be caused by, and we're still dissecting this apart, smoking, poor diet, metabolic disorders, exposure to environmental toxins, and other factors that can change the balance of those molecules in the seminal fluid. Anything that increases inflammation is likely to be bad for the immune response, the generation of the immune tolerogenic molecules in the male, and also the immune tolerance response in the female. So the things that we know are linked with infertility, obesity, poor diet, unresolved infections, stress, psychosocial stress, poor choices about lifestyle and exposure to various drugs and smoking, and exposure to environmental chemicals. All of these increase inflammation and impair this generation of immune tolerance. There are also various conditions in women and probably also in men, but we know less about it in men, that are linked with poor generation of tolerance for pregnancy, especially pre-diabetes and even diagnosed diabetes, so glucose intolerance is important, but all the usual candidates listed here that generate inflammation interfere with tolerance. So this then raises the question of whether we can predict who might be at risk of problems, and those people who've experienced the IVF system will know that there's lots of discussion and debate about immune diagnoses and immune tests. But the message I want to leave you with today is that none of them are any good. So to date, unfortunately, we don't have enough information to offer really good value tests to work out who's at risk. There are many clinics around the place that are offering to test your natural killer cells, but I've told you that natural killer cells are actually good guys, and having enough natural killer cells is important to get implantation and placentas operating correctly. And the Treg cells are likely to be more informative, but we don't yet have enough detailed information, especially how to work with blood T cells to get the right predictive tests. The other important take-home message is that turning off the immune system is not a good idea if you want to do that to improve your fertility, and many clinics are offering to administer corticosteroid drugs to suppress the immune response, and they're coming from the position that this might suppress immune rejection, but there is no evidence that this is useful, and indeed there is evidence that it's harmful. So at the moment, for unselected women, it's a bad idea. It increases the rate of preterm birth and of congenital abnormalities in offspring, and it's the only women who might want to use prednisolone and corticosteroid drugs are those who've been clearly diagnosed with an autoimmune condition. And that's because these drugs, they might turn off that immune rejection part of your response, but they also turn off, indeed they do more turning off of the immune tolerance part. So I think the future does hold a situation where we will have better tests for the immune response and where we'll have better treatments based on the immune response in pregnancy. And there are many scientists around the world that are working on, especially understanding these seminal fluid factors and different ways of driving the immune response towards tolerance. And some of the approaches are listed here. And I think within the next 10 years or so, we'll be getting closer to that. But for now, the message is that you need a strong immune response, and this is the best way to benefit your pregnancy chances, to be healthy, to have enough sunlight, enough vitamin D, enough protein in your diet, all the things that give you a strong immune response. And it's important not just in intending mums, but their male partners as well. The immune response to pregnancy has evolved for a good reason, for individual and population benefit. A strong immune response will boost the chances of reproductive success. Our treatments are evolving, but we're not there yet. And as scientists always say, we need more research to get us there. But in this case, I think it's looking promising, and we will. And this work is done by people all around the world, but most of the examples I've given you today are from our lab, and there's lots of great young scientists who've contributed to that work. Thank you very much.
Yes, thank you very much, Sarah, for an absolutely fantastic and inspiring lecture there, and sort of some fantastic images and some really challenging concepts that she's raising there, and thank you for posting with the left-hand side, for which I apologise. It's now open to questions that anyone may have for Sarah. Yes, sorry, yes.
For example, which kind? Yeah.